Dieses Meeting wird aufgezeichnet.
Here we go. So we're looking forward for a great presentation of the XtraTracks 2024. Actually, it's the first presentation, one of the really interesting ones, going rather to the domain of image processing, medical image processing, maybe a smart application in the field of rare genetic disorders, phenotyping. So today's speaker is Hsieh, which I definitely pronounced totally wrong, but maybe you could correct me, Hsieh.
Okay, great. Thanks for having me today and this great introduction to my presentation. And so my name is Hsieh. I'm currently the postdoc working in the University Hospital Bonn, and my PhD was mainly focused on this facial analysis for diagnosing the patients with rare Mendelian disorder. So we often call this kind of technology is called next generation phenotyping. So today I will talk about two main topics. One is how we utilize the facial analysis to diagnose the rare disorder. And the second topic is the data set we use in this kind of technology. So maybe first of all, we start from maybe you might wonder why we can diagnose the patients with rare disorder by looking at the face. So these are the three patients with the syndrome called Cordelia de Lange syndromes. I believe maybe you already see some patterns presenting in their face. So maybe I wait for maybe 5 seconds. Okay, so originally if this is the in-person meeting, then I would say if someone know the answer, I will give him or give her the Haribo from our city. Yeah, so I believe you already know the answer, which is their eyebrow. So the eyebrows in these patients, they all have a symptom called synovitis. So sometimes they have very thick eyebrow and even the eyebrow will be connected. Yeah, so this is just one of the key features presenting in this disorder. So we know that with this kind of facial features, we can try to diagnose the patients with recognizing the facial patterns presenting in their face. Therefore, what we are doing here is we just simply train some deep learning convolutional neural networks by thousands of patients' photos. Then we can train a network, try to learn the facial representation of the disease. And later we can just take the photo on the left-hand side from the patient, and then we try to quantify some similarity score or some probability to the syndromes we train in the network. So for example, if today these patients have a very high similarity score to cofencyrus 1, then we can further check the disease gene or disease-causing mutations related to this disease, and then try to find out the variance in patients' exome data, and in the end try to find out the answer of this patient. Yeah, and this kind of technology, we often call it like next generation phenotyping. This is the same idea, try to echo like next generation sequencing. So we think this kind of computer vision, and it really drive the advancement on this field for the rare disorder. And so when it starts, so it starts from like around 10 years ago at the very beginning in a paper published by Ferry et al. In eLife in 2014. So they start from this kind of training the network or training some models on patients' images. They start from a scale with around 1,300 photos with only 8 syndromes. This is around like 10 years ago. And after 5 years, so the company, there's a company called FDNA. They have a online platform called Face2Gene on the right-hand side here. So they published a paper called Deep Gestalt, the method called Deep Gestalt. I will refer to this name in the following presentation. They published Deep Gestalt in Nature Medicine in 2019. So in this paper, they trained the deep convolutional neural network on over 20,000 images with around 200 disorders. So after 5 years, we finally push the scale from 8 syndromes to over 200 syndromes. And however, this kind of technology still have some bottleneck. So for example, on the left-hand side, they can use some different facial region, try to classify different genetic disorders they train in the network. However, because we are analyzing the rare disorder, so there are around more than 7,000 or 5,000 disorders in we currently know. So, but with this kind of technology, we can only diagnose around 200. So it is far less than what we need in the real world scenario. So that's why in 2022, so we and this company FDNA, we work together. We further publish another paper called Gestalt Matcher. So we published this work in Nature Genetics in 2022. So what we do here is we are taking the feature vector from each patient or from each photo, then we try to quantify the similarity based on the feature vector. So instead of training a model and only predict the disorder, we train in the model. And with this kind of transition, then we can try to support over 1,000 disorder. So it's far larger than originally around 200 disorder. And this kind of transition also enable us to analyze on the patient level similarity, because sometimes we are not just want to predict the disorder, we also want to know whether these two patients are similar or not, because it can help us to find the second patient of this disease in the world. Because sometimes maybe your patient is the only one reported in the world, then you want to find the second one. Then we can utilize this kind of technology, try to identify another patient with the same facial features. So maybe we would like to start how we do it, because we are working on like the rare disorder. So it's super rare. So sometimes there's only 2 patients in the world for some disease. So we don't have a very large data set, just like normal computer vision task. So you have like millions of images you can train. So we have only around 10,000 images. So what we do, what the deep Gestalt this paper do at the very beginning is just they are utilizing the transfer learning. So first of all, they train the deep neural network on the healthy individuals from the Hsieh web phase. There are around 500,000 healthy individuals. So like Matt Damon or Tom Cruise or Mark Wahlberg. And we are trying to, so this network, we're trying to find out how we classify different persons in the first round. So for example, this is Mark Wahlberg, and on the left-hand side is Matt Damon. So I feel they are really similar. So I finally know that they are different person until I got my PhD. So it is really difficult task, I would say. So first of all, in the first round, we train the model with the healthy individual. We try to learn the feature vector to classify different facial feature to classify different person. And then we replace the last layer from different person to different disease. So for example, here we try to learn the disorder like Cohen-Cyrus progeria syndrome, or the last one will be Cornelia de Lange syndrome. Then we further train on more than 10,000 images with around 200 disorder. We try to learn how the facial features can classify different disease. So with this kind of technology, then you can further to try to classify the disorder. You train on the last layer, which is the classify layer. So this is how the deep Gestalt was doing in 2019. However, what we have, and this kind of technology is also already very well known in the market, which is called face-to-gene, this platform. So a lot of clinicians, they just use this platform and try to take the photo and try to diagnose the patients based on this platform. So this is what they provide over the last few years. And this kind of technology still have some limitation because we are working on the rare disorder. So for example, if you want to train a disease in the models, for example, here we only have 4 diseases: Cohen-Cyrus, progeria, and Cornelia de Lange, these 4 diseases. If today you want to support another disorder, then what we need to do is like we need to collect enough photos for this disorder. And also today, if you only you train on the fifth disorder, maybe tomorrow you want to train on the sixth disorder. So this is not very flexible. And also we are working on the rare disorder. So sometimes you are not able to really collect a lot of photos. So sometimes you only have 6 or 5 patients, or maybe less than 5. This is very normal in this in this context. So therefore, what we want to do is like we want to make the whole approach much more flexible to support the novel disorder or to support the super ultra rare disorder. So what we do here is like we take the last fully connected layer, which is the blue ball here, as a feature vector. Then we use the feature vector to span another phenotype space. We call it like clinical phase phenotype space on the button. Then in this space, each photo is a point located in this space. And then we can further do some clustering analysis or try to match the patients with some distance functions calculated in this space. So for example, we will see like Coffin-Cyrus patients will located nearby, and also the Collinia de Lange patients will also cluster together in this space. So, and this kind of extension, we call it Gestalt measure. So instead of only classifying the patients in the classifier, we trying to use this kind of feature vector and try to span another space and do more downstream analysis. So then this is the key difference between these two different approaches. So in this space, how we quantify the similarity is that we utilize the cosine distance to quantify the two patients' similarity in this space. So for example, if we know that if they are close in the space, which is on the left-hand side, if the patients are clustered together, then we know that they might have the similar phenotype. So for example, the Coffin-Cyrus patients will be nearby. Noonan or Nicolaides Brahe-Zeit syndromes, they all will form another cluster. And so the previous Gestalt measure will be our is more like our proof of concept on this kind of technology. So later we try different kind of techniques and try to improve the performance. So what we try, the first try is called model ensemble and test time augmentation. So what we do here is we try to, so for example, we try to do the test time augmentation. So we try to do some flipping or coloring, and we have 4 different versions of the photo we can test in the test time. And we also train 3 different models. We try different kind of regulation and also different kind of architecture. We try to have find out more balanced way to train the models. So in the end, we simply just average all the distance among these 12 different vectors and try to find out the final distance. So this is what we published last year in the Winter Conference of Application of Computer Vision conference. So we try to propose this kind of technology can further improve the performance of the prediction. And then you might wonder, so then how we evaluate the performance? So here we use the top K match or top K accuracy to evaluate the performance. So what we do is here like this. So on the right hand side is our gallery or the space with a lot of patients you can match. And what we are doing now is like we are testing the patient on the right and left hand side, and the correct diagnosis is the Coffin-Cyrus. We already know the answer. And then we try to find the most similar patient or nearest patients in the space on the right hand side. So if the nearest patient is Coffin-Cyrus, then we call it top 1 match. And today, if there is another patient which is have the Noonan syndrome as the disorder, and now we also test this patient, and we also put this patient into the space, and then here we found that another Noonan patient in the space is in the 5th position. Then we call it top 5 match. Then it's this kind of evaluation also apply to top K, top 10 or top 30 match. Then we just trying to evaluate how much percentage of the patient can reach top 1, top 5, or top 10 match. then try to evaluate the performance in this way. So before I report some of the accuracy to you, I will first of all try to introduce the extremely biased distribution in this kind of this kind of field. So for example, this is the distribution of the disorder in the data set we have. So the x-axis is the disorder, how many patients this disorder have. So it's from like 2 patients to 350 patients. And the y-axis is how many disorder have this number of patients. So for example, here we see on the very right-hand side here, there is one disorder with more than 300 patients. And there is another one disorder with around 200 and 250 patients. And here we can see that most of the disorder is all squeezed on the left-hand side, which is less than 50 or less than 10 patients. It just reflects the real-world scenario. So the relatively frequent disorder in the real-world scenario is very rare. So most of the disorder, you only have a very, very little number of patients, which is on the very left-hand side. Then we call this kind of set below 7 patients, we call it rare set. And if the disorder have more than 7 patients, we call it frequent test. So this is just two different definitions we will use in the following slide. So maybe first of all, we wonder, so for example, in the very previous slide, after the metademon slide, and we said that we will use the Hsieh web phase to do the model pre-training. So here we trying to try different pre-trained data set. So we try VGG phase 2, VGG2, Hsieh, and also like the Glean's 360K, which is have around 350,000 patients, or not patients, sorry, 350,000 individuals. So we are trying to see whether different kind of pre-trained data set can influence the prediction accuracy. So here we can see that at the very left-hand side, if you increase the size of the pre-trained data set, you will have a very significant improvement of the prediction accuracy. However, from this MS1 MV3 to Glean's 360K, although we increase almost 3 times larger the data set, but the performance increase is relatively small compared to the left-hand side. Then we know that it's not like you always increase the data size, you will achieve a higher performance. And then we would like to show you some, you might wonder, so how accurate or how the performance of this kind of technology is in the data set. So here I will show you a very relatively very ugly table. And so here I would like to report different performance based on different technology and also different experimental setup. So for example, in the first row, which is GN Hsieh 2022, this is what we published in Nature Genetics, which is the first time we published the Gestalt measure. And on the middle side here, we have R50, R100, and the other setting. So R50 is the ResNet 50 and R100 is the ResNet 100. So we are trying to use different architecture to see whether we can increase the performance. So first of all, we can see here. So the first comparison, we can see there's a huge difference if we apply the relatively latest architecture, which is ResNet 50, compared to what we have, what we used in the previous publication, which is the architecture we used like 10 years ago. So here we can see there is a significant improvement in the top 1 accuracy, almost 30%, and top 5 accuracy is also 30%. So therefore, we know that so in the previous publications, our architecture maybe is not really so suitable for this task. So we already find our ResNet 50 or ResNet 100 can significantly improve the performance. And then in the end, I would like to introduce is what we do, the model ensemble. Model ensemble, what we do is we select 3 models in in the middle. So we try to ensemble the three models together. So one model we select, which is achieve the highest frequent set performance, and another one we will select, which is achieve the highest rare set performance. In the end, we also select one which is only seeing the healthy individuals. So we're trying to use this way to average the performance to support two different set of the disorder. And in the end, we can see that so we improve the performance from 15% to almost 52%, which is all more than like two times of the improvement. So then therefore, this is just a example how we can improve the performance. So we are not saying like this way is the state of the art of this technology. So but because this is the first data set of first approach we can use the data set that is open to public. So that's why this kind of example is very meaningful in this field. So another thing I also briefly talked at the very beginning. So what we can use is also we call it Gestalt matcher, which is we call it like a facial image version of the gene matcher. So maybe I introduce a gene matcher slightly here. What the gene matcher do is like you can input some mutation or some clinical feature, then you can try to find another patients in maybe another side of the world. Maybe you find it a patient in Australia, or you find a patient in maybe Brazil. By this kind of matching mechanism, you will receive the contact of the clinicians who have this patient. So therefore, we imagine like Gestalt matcher can also achieve this kind of um, a task. So for example, before, usually how the doctor find another patient is very old school. So they all go to some meeting conference and show the photos to everyone. Is there anyone have the similar patient like this patient? Then this is the way how people find the second patient. Therefore, this kind of platform, this kind of technology will be very helpful for them. It's like they only need to upload a photo, then maybe after few months they will get a notification because there is another very similar patients to your patients is uploaded to the system. Okay, so to date, this Gestalt matcher is already used to analyze around 19 novel disease, and 15 of them already published in the peer-reviewed journal or medRxiv, which is the preprint server. So therefore, this is how can be used not only for the diagnosis, it can be also used for analyzing some facial similarity of a given cohort. And so it is also very helpful for the clinicians or clinician scientists to publish some paper. Okay, so I will try to move to the second topic, which is the database we use to train this technology. So before, I would like to start from a small story. So for example, the reason why we want to set up a database is not just want to develop our tools. We want the training data. Another reason is just like this use case. If today I'm analyzing a patient, which is on the left-hand side, and this patient have some clinical feature and abnormality of the eye, red green. So he has some problem with red green distinction. So and then I found that okay, I want to compare these patients to another disease, which is Cornea de Lange syndrome. How do I do it? And what I will do next is like, okay, I will try to search the literature, try to find out the photos. So for example, this is one publication, and then this is another publication and a lot of photos and a lot of labeling I need to do. And then I need to go through a lot of tables like this, which is extremely difficult to do. It's difficult to read, and I had to type everything one by one and into another format I will use in my research. So then you need to do everything yourself, and you need to redo everything like this again and again if you want to do another analysis with another disease. So therefore, we feel this is too laborious, and we are not able and it is not able to scale and also reproducible because everyone might use this kind of information differently. So therefore, what we do is that we build up a database, and so many medical students and also the clinicians help us to start this project. So in this project, we will store the facial photos and also the other type of medical images. So like what you see here. So then the clinicians, like the clinician want to analyze the previous patient, they can simply come here and to see how the patient looked like. And then these patients are also well labeled. So for example, they label with uh, the age when they take the photo and what is the disease causing gene and what is the symptoms they have, and also, for example, and also very important, where is the source? So is this the unpublished patients from some clinic in maybe in Germany, or is this some patients already shown in some publication? Then we need to cite the source. Then this is how this database start. And this database we call it Gestalt Metric database. So currently we already have around 9,700 patients in this database, and so as I briefly mentioned, like many medical students and clinicians help us to read the paper and do the annotation. So we already annotate from more than 2,800 publications over the last around 3 to 4 years. And we already cover almost 800 different disease, or almost 900. Yeah, and this platform, Gestalt Metric Database, is hosted by AGD, which is the nonprofit organization in Germany. Then so we have a committee board that will review who can do the download, who can access the data. So, and to avoid that, not the research coming, some people who are not doing the research and can access our data set. We will try to avoid this kind of situation because the patient data is relatively sensitive. We don't want people randomly download the data and distribute in some web page, and we are also avoid that this can be indexed by Google. So if you don't have account, then you are not able to access from Google. Yeah. And so this is just some overview of the design of our database. So what at the beginning, first of all, we start from the publication. So many our colleagues read the publication, 2,800 publication, try to annotate the whole database. And we are also inviting the clinicians, physicians. They will also invite directly the patients they know to and also their parents will also contribute us the data, so then they can sign the digital content directly in our platform. Then they can contribute their cases. So in this data set, as you can see here, we have the photo, the portrait. We also have the fundoscopy images, like the hand X-ray images and MRI images. We also store some metadata like the sex, ancestry, age, and also different kind of medical condition. So in the end, we want to provide this kind of technology to the users who is the maybe some medical member, medical faculty, some clinicians, and then we also want to provide the computer scientists to access, because computer scientists have maybe a lot of novel way to improve the deep learning technology, but they don't have the data. That's why this kind of research field is relatively far behind the computer vision field. So that's why we would like to provide this kind of data set to the computer scientists. Then they can try to benchmark to develop the method based on the same data set. And in this data set, we provide one different functionality. So first of all, we provide a search. So for example, you can search some disease, and then you will see all the faces with these disorders. And then you can also do some analysis. So you can analyze the case of your analyze your face with all the data in our data set, in our database. In the end, will be the purpose for the computer scientists. Then they can try to train the network with our data set. And also, we also provide some diagnosis platforms so people can upload a photo and try to get a diagnosis. Then this is what we recently developed, which is we just announced it, released it last month. And we also provide the REST API can be connected by some hospital. So they just send the photos through the REST API to our server. Then we will return the results to by REST API. Yeah, so this is what we can provide through this platform. And this is just an overview how the patient really looked like. So for example, on the left-hand side, upper left corner, we will have some citations, which is where this data come from. Then we will have the case ID, and with some meta information, we will store the photos at different ages. This is very important because sometimes the face will evolve through the times. So sometimes you don't look very similar to maybe 20 years ago. And also the facial dysmorphic features will also evolve through the times. So that's why this is also very important for us to know, to have the photos at different ages. We will also store some genetic molecular information and the phenotypic information. And also you can see here, we also store the photos, frontal photos, and in the middle is the profile and also the hand. Because sometimes not only the face is meaningful for the prediction, and also sometimes your limbs and your foot or the other part of the body is also very, very helpful for the clinicians to find out the answer. Yeah, so just like I briefly mentioned at the beginning, so we start from looking at the publication, and this is the first stage. However, we encounter some bottleneck because sometimes people don't publish some relatively frequent disorder anymore. However, we still, as you can see in the distribution, very beginning, like many disorder, we only have around 20 or 30 patients, and you don't find any more in the literatures. So that's why we start this worldwide collaboration. So we start contacting all the clinicians in different countries. We try to recruit the patient directly from their clinic because they might have the patients in their hospital. They just don't have time to publish as medical literatures. So that's why the photos hidden in their computer, and they also have the consent to publish. They just don't have time. So that's why we start this collaboration with all of them. Then we collect the data directly from them. So until now, already more than around 300 clinicians already contribute some patients to our database, and we already collect around almost 1,000 patients. And if we count the images, we already have more than 2,400 images, which is not published in any medical literatures. And we also collaborate directly with the patient support group. So for example, there are some disease, they have some patient, we will meet the parents, and then some of them are very really interested in this kind of technology, because if we have more photos, larger data set, then we will have a higher performance of the prediction. It might help in the future for the other parents have the similar situation. Similar situation to their children's. So because it is meaningful to have an early diagnosis, because sometimes if you can diagnose the disease earlier, you might have a proper treatment at the very beginning. And then you might have some you might wonder, so how the data really distribute in our database. So here you can see, so the sex is relatively balanced. So we have around 48% are male and 44% are female, although the male are slightly more than female. But I would say it's still relatively balanced compared to the other distribution. So the second one we might want to see is the age. So because usually the rare disorder is diagnosed when they are relatively young, because the symptom or all the disease will already develop at a very relatively young ages. So here we can see that the majority of the data coming from the age below 10. So there are more than almost 70% of the data coming from the age below 10. So here we can see that when the patient are still baby below 1 year old, they are around 13%, and 32% are the age between 1 and 5 years old, and 23% between 5 to 10 years old. So it tells us like the data set is the model is mainly trained on relatively young age. So it also might have some bias in the training because it might have the relatively poor performance for the patients who is relatively older. And we will also report this the accuracy later in the following slide. And then it comes to another very important distribution in our database is also which is the ancestry distribution. So here we have around like 19%. We don't have the ancestry information. So here we just leave it out. We don't talk about this unknown category. So here we can see that just like all the data set in the medical field, the European ancestry is always the dominant the whole data set. We have around 67% are from the European ancestry, and 19% from Asia, which is Asian, and 7% is from African, and around 7% is others. So in others, we have American Native and Native American, and also the Latin American. So here we can see that there is a huge bias in the data set. So that's why always a lot of clinicians from non-European countries, they always asking this question, is your technology useful or helpful for my non-European patient? So then this is what we would like to also want to answer in the following slide. So first of all, we already see that there is a huge bias in the data distribution. So most of the data coming from the European countries or a European ancestry. So what we are doing here is like we try to contact the global clinicians. We want to collect the data directly from their clinic. Here we call this kind of cases called micro publication. So refers to we collect the data from the clinician directly. Here we can see that so the distribution here, the darker color here is directly from the clinicians, and the gray color, light gray color is already published in some medical literatures. Here we can see that in the African countries, almost 40% of the data is coming directly from the hospital or directly from the clinicians. If we break down to more specific subcategory or subgroup of the ancestry, here we can see that even more like a sub-Saharan, this subcategory of the ancestry is even more. We have almost 80% of the data coming from the clinic directly, which is these data are not published in any literature before. It tells us that this kind of collecting or recruitment strategy is really helpful or beneficial for this underrepresented population like African countries. So therefore, we believe this is the way in the future we can improve this data distribution to have more relatively fair distributions regarding the ancestry. And then you might wonder like, okay, so whether the performance is different between different sex. So here we can see that so the column is the top 1 to top 30 accuracy, and the different row is the male, female, and unknown. We have some patients we don't know the sex. So here we can see that the male and female, they achieve relatively very similar performance. I would say is is quite comparable, although still in top 10 accuracy, still around like 2 to 3% difference. But I would say it is still relatively comparable. And then the next things we would like to analyze is how the age influence. So because some people, some doctors is really interested to know like whether we can diagnose the patients when the patients are still a young baby. So this is also very important because some of the disease will develop or very important. You need to have the proper treatment when they are still like the baby. So for example, some liver disease, if you can give the treatment earlier, then you can avoid some liver transplantation. So that's why we are also interested in the accuracy below 1 year old. So as we can see here, so as we expected, so the baby is very relatively difficult to recognize. So they achieve slightly lower performance, like maybe around 6 to 5% performance difference compared to the other age group. And we can see, I would say, the age between 5 to 10 years old, the performance is relatively higher and relatively stable. Yeah, so, but this is something we will need to dig into more when we have more data from the patients with different ages. So in the end, we would like to also want to answer the previous question is like whether the underrepresented population really achieve the lower performance. So here we also break down the ancestry group into African, Asian, European, and others and unknown. So here we can see that in the test set of 882 images, we can see that we didn't see African and Asians really perform worse than European. So, for example, the top 5 accuracy, African achieved 82%, which is 77% higher than European. And top 10 accuracy, the Asians achieved higher than European as well. However, we found that the problem will be the data distribution, because here we can see that European have around 500 photos in the test set, African only 29, Asian 100. And here we found that the European cover much more disorder, which is relatively difficult to recognize. So therefore, this kind of comparison is not fair if we compare the performance directly, if they are not analyzing the same disorder. So therefore, we further do another analysis. Is like we only compare two different ancestry on the same disorder. So for example, we compare African and European, which is the first row. We only analyze on 6 disorder, which is the overlap between these two groups. And also on the second row is the Asian to the European, is the 36 disorder. So here we can see that the European always outperform Asian and African, and also I would say very significantly outperform European will outperform Asian and African, which is what we expected. And this is also what we need to improve in the near future. So how we can improve it? So we would like to prove that the next question is like whether the more diverse data in the training set can improve the performance for the underrepresented population, like Asian and African. So here what we do is we train 2 different models. So first model is the red models, which is not red, sorry, blue models, which is we only train on the European patients. So the blue bar is only trained on the European patient. And this yellow bar, which is trained on European, also on the non-European patient. So so to simply explain it, like the blue bar only see European. They never see the other ancestry in the training set. And the yellow bar is like they not only see European, they also see the other ancestry, like Asian and African. So first of all, we see that we compare the performance between these two models. We can see that when we are testing the European patient, the blue model will outperform the yellow. Yeah, which is because the model is already fit extremely well on the European. because that's why they perform better. However, if we now we look at the yellow bar and on the sub-Saharan, for example, if the models already see more diverse data set, then the yellow bar will outperform the blue significantly. It also applied, it also applied to the other ancestry, like Asian or the other African ancestry. So therefore, we trying to prove that improving the diversity in the training data can really improve the performance, especially for the underrepresented population. And also we see that the performance drop down for the European is really slightly. So it also tells us that originally our model is fit too well, is very biased to the European ancestry. Okay, so we almost come to the end. So this kind of data set is also very important for the next generation phenotyping development. So the reason is because originally, before Gestalt measure database came out, there's no public data set people can really develop this method in this field. So the data are all owned by the company FDNA, which is the company provide the service. So that's why no research group can really develop this kind of technology. So as we think this is not very healthy in this field. So that's why we make this data set as a fair data set. So the data set can be findable, accessible, interoperable, and also reproducible. So then one of our collaborator, they also try to integrate the facial image analysis with the large language model. It means that you can just like open your ChatGPT, upload a photo, and write down some clinical feature. I have some feature, blah blah blah, and then you will try to get the diagnosis. So then this is the first application that can integrate the facial photo together with the clinical text. And so we envision this can be very impactful in the future. So yeah, so this is just some future vision in this kind of technology. So in 2022, we try to push the boundary to more than 1,000 disorder, which is by our Gestalt measure paper. And then later in the same year, Dawn et al. From NIH in the United States, they also work on different direction in this field, which is they try to use the gun model, style gun, to try to generate the synthesized face of the patient. So because as we know that the faces is really sensitive. So some patients don't want to share the photos because they don't want to be recognized in the society. This is especially very challenging in the Asian countries. So that's why we cannot really collect the data from Asian countries. So therefore, this is the first paper to validate that we can utilize the style gun, this kind of approach, to generate the the fake patient's face, but they still look like a real patient. Yeah, and afterwards, until now, we envision with this kind of data set, we can play more, we can dig into more into this field. We can analyze how the age influence the prediction, and also whether we can synthesize the patient's face with different ages. And also we can analyze the ancestry and also the facial expression. And facial expression is also very, very important when you diagnose the patients, because if the patients have a happy face, like laughing, sometimes it will influence the prediction. So for example, some disorder will always have some facial expression, so like Angelman syndrome, so the patients are always laughing. So then it will lead to some bias if you want to make it as an application. And also in the end, it's the explainability. So and this is a very huge problem now in this medical field. So we all use this kind of deep learning to predict the disorder, make it like a medical device, but no one can really explain like why the model tell us this is the answer. So you told me this is the model told me like this is disorder A, but we don't know why the model think this is disorder. Is it because of the eyebrow or because of the nose or mouth? So this is what we need to solve in the future if we want to really make this kind of technology happening in the hospital. Yeah, in the end, I would like to thank all my colleagues, also the colleagues Tori, and so we worked together since almost 7 years ago. Yeah, and also all my colleagues in IGSB and Charité Berlin, and I also want to thank the patients and also their family. And thank you. I'm happy to answer all the questions.
Yeah, thank you, Chung-Ching, for this great presentation. And I think indeed Jean, Tori, Pantel will join us for the discussion,
Right.
as she is as well co-author, organizing mind in this research. So we have two researchers in the field available for questions. Are there any questions from the auditorium? Otherwise, I would start with one because I have few. And I would go from the end to the beginning in a way, like you presented really an interesting approach, deep learning based, to identify genetic rare diseases in patients based on photos. And you said like it will be challenging if we can't explain how the methods are working. Have you considered so far to apply like explainability approaches, interpretability approaches, and maybe do you already have insights what would be the right resulting accuracy? So there might be a loss of accuracy in comparison. Have you investigated to that? And
Yeah.
yeah.
Yes, thanks for this is a very good question. So yeah, we do try some very different kind of like the explainable AI stuff, like a GradCAN or many different kind of this kind of similar approach, trying to identify the key regions result into the prediction, like whether disorder A and which facial region activate this disorder. However, I think this is one thing can use for some debugging or some to understand whether there is some problem with the face. For example, we are analyzing the patient's face. So sometimes there are some tubes on the face because they are still in the clinic. So sometimes the tubes on the face or there are some components on the face. It will also influence the results. Maybe this lead to the prediction. And another problem will the true challenge now is like even though we highlight the eyebrow, we know that eyebrow is very important for this disorder, but we still don't know what happened to the eyebrow. Is the eyebrow too thick or too thin, or is there's no eyebrow or not? So there is still no this kind of explanation from the model. We still don't know how models think the eyebrow. So that's why we feel we need to provide two different output. The first output is the visual output, is like which region is important. And then maybe model can output us some explanation, like some text, like synophrys, thick eyebrow, no eyebrow, and so on. This is what we are still working on.
Yeah, and that's a good bridge then over to foundational models, maybe even. Huh?
Yeah.
Yeah. Yes. Thanks a lot. Any questions from the others?
Andreas, I would have a question which goes into the same direction. So if you start with the whole thing, I mean, that's quite a while ago, I guess, but how do you define your initial let's say design of the network? I mean, we've seen that it's like 10 layers and then there's this matching layer as an 11th layer, and how many nodes do you take, and all of this basic stuff, right? There's millions of things you can decide on. I mean, not millions maybe, but a lot, I guess. How do you, and I mean, you have your goal, of course, so it should recognize and distinguish between these different diseases, right? But how do you start as a kind of how do you start with the whole effort, right? I mean, because there are so many possibilities how you could do it, right?
Yeah.
Or you have so many freedoms, let's say, how you could do it.
Yeah. So there is a huge story behind it. So at the beginning, we don't have any choice, or so for example, we know that at the very beginning from this phase 2 gene or FDNA, this company, they have everything. They have the data. So you need to have the data to start the training or to start in this field. So they take a paper called Hsieh web phase, which is this 10 layers. I forgot really exact numbers. 10 layer, which is very extremely classical neural network published in 2014. So they take this network and the pre-trained data set and to publish the paper in 2019 in Nature Medicine. So the reason why we take this same architecture and also the pre-trained data set is because we want to have the fair comparison to the paper in 2019. So that's why we take this relatively outdated architecture. So as you pointed out, like, can we do some benchmarking with different technology, different architecture, different maybe foundation model and so on? But because at that time, like 3-4 years ago, when we work on this Gestalt project, we are not able to access the training data. So this is the reason why we are not able to really play it. So because this is the property of their company. So this is one of the reasons we start this Gestalt database, because then in this world, the other research community can, a research institute can also work on the same question. People can try different kind of architecture. So that's why in 2022 we try different architecture, ResNet and so on. We also play like ResNet 50, ResNet 100, and also the other like loss function. We're trying to optimize whether playing this kind of different technology can improve the accuracy. So but we are still in a very early stage. So so this is the first publication we published last year. We tried like ResNet and we didn't try the others yet. So we would like to leave this question to all of our collaborators because many of them are trying. So one of them are trying this transformer to see whether they can try to integrate the image and the large language model seamlessly together. Yeah, so this is
Thank you.
some story behind.
Yeah, it's always interesting to hear those stories. Thanks. Yeah.
Okay.
Go ahead, Nico.
Maybe I can ask a question. At the end, you talked about clinical translation and these kind of issues. So how about, let's say regulatory issues? I assume that if you want to put it into clinical practice, you would have to make a decision if this is a medical product.
Yeah.
Did you think in this direction, and what's your impression, or how do you want to tackle this?
Yeah, so this is a very good question, and this is also exactly what we encounter right now. We are trying to make it like a medical device, and then they said one of the regulation, like you pointed out, you need to have this explainability. So this is also answer the previous question, like we want to provide this explainable AI stuff, like the visual explanation and also some text explanation to really tell the doctor how the model think. So maybe Tori, you want to?
Yes, I might add the solution we found at the moment at the university hospitals is ongoing research, and we have ethics, ask the ethics committees at different locations. So that's the way it works now, and we're using it in clinical routine as every case is also a research case in university. Yeah, that's now our way to go, but the goal is to have it as a medical product, yes.
So now I think lots of clinicians use it every day, but we just cannot say this is the medical product or medical device. Just give them some advice.
Yeah, I understand. I completely understand the problems that you face. But yeah, good luck with that. I keep my fingers crossed that you will make it to an actual product. That would be really great.
Thank you.
And I mean, I like the approach to say, like, we make a matcher, we match to the next similar cases. So the decision taking at the end is rather at the doctor. So you don't suggest the one disease, but you rather have support tool suggesting which disease could be relevant or could be a fit, right?
That's something I would like to add from a clinical point of view. The diagnosis is made by sequencing, right? You sequence the genomic data and look into that data, and the image data is just an add to help filter out variants and to prioritize the variants during filtering. At the moment, we clinicians sit there, go through the variants one by one and do a lot of manual search. So that is just another input for interpreting variants, especially in times of genomes.
And I mean, listening to your talk, it could work as well as a pre-screening tool, right? Where in case every kid born at a hospital directly gets made a photo, and the photo is directly pre-screened, and then you rather might want to have a binary screening, which says, oh, there could be a rare disease or there could be none, right? And then maybe in that approach, so that would be the question. Have you evaluated such a binary classifier as well, and what would be the classification accuracies in that regard?
Yeah, Tori, I think you published a paper on it, right?
I published a paper on it. Yes, we did this with the data from the from Face2Gene in the first round to have a validation there. And to what we did is use the vectors of the first 30 matches of this algorithm and put them into a support vector machine to have put it on top, because there is no healthy class. So it's always like in syndromes that not only the dysmorphism of the face is the deciding thing, but also there are other symptoms of the patient, right? That's something to say. So just because someone looks a bit different, it isn't a hint for a syndromic background. So, and what we published there was, I tried to, if I can really quickly open the paper, but I think it takes too long. So what we did, we had a control group with images of patients that were, or individuals that were matched by age, ethnic background, and sex, and we could separate them with the area under the ROC curve of 0.73. That was the best in the three tools we compared. And with the support vector machine on top, we could improve that to 0.86.
So you will only panic around every 8 patient or parent without reasonability. So that's a good value, maybe. If it's okay, maybe you should go to a doctor to have additional audition like screening, right? Yeah.
Especially in regions where the genetic department is maybe more far away than in other regions, yes. But still, there is no healthy class. It's more like we give some kind of advice if there's, for example, a high difference in the first 2 matches, in top 1 match and top 2 match. It gives more a hint that there is this syndrome is present than in others if there is a low score for all of the top 30 cases.
Mm-hmm. Have you, I mean, Cheng is from University of Bonn, and you, Jean-Tori, are at least have been at Charité, still
right.
at Charité. So there are two main hospitals associated in the Medical Informatics Initiative. So I assume both have rolled out like the broad consent, which allows the use of signed use of most medical data. Have you discussed in the hospital context if it's possible, for example, to make an initial screening photo of the patients and use that, or to use existing early photos even though the genetic disorder has not been diagnosed? Do you have any experience in that regard?
So we were talking with the Centers for Children, or what it's called, Sozialpädiatrisches Zentrum, Social Pediatric Center, because they would see young children that are somehow have some medical difficulties, but at the moment it's mainly at the medical genetics departments, because there's still the high expertise. But that would be a next step to integrate it on a more basic level.
But
we didn't do that yet.
Okay, and so another great advantage in the context of the XtraTracks might be that you're really globally organized, right? So your data set is like a global data set, and so you have experience with the different healthcare systems. So how challenging is it for you to get, for example, data from Asian clinics? So is it then rather the medical professional which takes the images, or are there standardized approaches and you get just the upload of this patients with the corresponding ICD-10 or 11 codes or similar?
Tori, you want to, or?
Well, it's very, very different from system to system to get the data. From Asia, it's especially challenging, I would say.
Yes.
So there was a paper some years ago of a Japanese group that had their own server in the country that had the model on it, but where they didn't share the data over the country's borders. We also have the option over the research ethical to have the patients directly share their data. So a doctor could send a link to the patient, and the patient can register their data and also change them after some time and add additional data and so on. And they can sign the data security form in the database.
Yeah, and thanks for Tori to point out that paper done by a Japanese group, because I feel it's interesting because I visited them last year in the conference in Japan, and I have this kind of discussion with them, and I asked them whether they want to join our study. Then they clearly told me like no one can send the data outside Japan successfully until now. So this is very challenging. And also, I'm originally from Taiwan, so I also talked to many Taiwanese doctors about our project and also this kind of facial analysis. They told me sometimes, even though they just only want to use it to diagnose the patient, the parents still say, no, don't take photos. So I don't know whether in Germany you have this kind of situation, but just they have this kind of social pressure. They don't want their kid be identified with some problem in the school. So
yeah, the spectrum is very different, because there are also a lot of families that are very happy to share the data and are very open about it, because it took them 10 years to find the right diagnosis and want to support future parents and their children to get a diagnosis sooner and maybe a better treatment through that.
Yeah, so it's very different from family to family.
So do you think there is hope in regard to data privacy? And if any one of the other participant has a question, just shout in between, because otherwise I have more questions. So data privacy, like anonymization of the images in a way that physiological features remain valid, or do you have any hope in that regard?
Yeah, actually, so one thing we want to do is utilize in this kind of GAN or diffusion model, try to synthesize the fake patient. Another thing is we are working with one of our colleagues in the hospital, not from directly in our groups, and they published the method called swarm learning in Nature like 2 years ago, and they already stopped this. They co-work with a company called HPE. They already have a product they can install directly in different clinic. Then they can start this kind of swarm learning or federated learning, this kind of stuff. And so without transferring the data, then they can train the model together. I believe this is especially very good fit to the Asian country because people don't want to share the data. So I believe maybe can be something we can achieve to overcome this situation in the future. Yeah.
Excellent. Yeah. Any other questions? In that way, what would you wish for? So what would be, from your perspective, the next big step you could achieve?
Yeah, the next thing, like you already bring it up, like we are already working on set up this kind of consortium. We want to enable training a model in different country, different hospital. But there are still some of the technical issue and also the political issue, like, and well, they are waiting to solve. And yeah, will be the first forthcoming challenges we want to solve. We want to more like connect the whole the consortium in different countries together to solve the problem of the diversity, because now still like a European dominant in this kind of data set. Yeah.
And my wish as a clinician would be that the GMDB is accepted as a micro publishing tool to have also patients give them possibility to update their GMDB profile, to have photos from different age groups, to train the models for more age groups as more patients are getting older than earlier because of better medical treatment.
Yeah.
So I think that's reasonable that you will achieve these goals soon. I wish you best of the luck on the way. And thanks a lot, really thanks a lot for this great starter to the XtraTrack season. Yeah, thanks Jean-Tori, thanks Chung Hyung for this great presentation. I think that comes from heart from all of us. And to everyone, see you again in 2 weeks. Where we rather have a wearable based approach, and then in 4 weeks we return to the vision phase again with data synthesization and extraction of some medical relevant parameters from images of users. So thanks a lot for today.
Okay, thank you very much.